Welcome back friends. Welcome to another video tutorial from Shomu's Biology. And in this video lecture, we will be talking about the pathogen recognition, which is one of the first part of innate immunity. Because remember, we are talking about the immune system of human body. And we're looking at a series of videos that demonstrates the immunology in more details. In the earlier videos, we talked about the overview of the immune system and how immune system is maintained and what is the importance of keeping innate immunity and acquired immunity and how they actually communicate with each other. Now we go and dig a little deep to talk about the innate immunity part, that is from the beginning of an infection and how the innate immunity help us to go against it. And this is the first part of the innate immunity lecture, and this is about how the cells of our immune system protect us against infection, how they fight against infection. Now one simple question before all this should be in your mind, that is how exactly a cell first identify as a foreign object. Because you know, the most important thing of immune system, not only to fight against infection but also to distinguish between what is foreign and what is self. And that is another very important feature. Now, so first they need to identify either the fragments of the bacterial body or fragments of virus that can be detected by cells of immune system. Once they detect those regions as foreign, then they can go against them to develop immune system. Now, if they fail to figure out which is foreign, then this foreign material can make us diseased, or if it gets its own material as foreign, that will also cause autoimmune response or autoimmunity, which is also bad. So in this case, we are talking about how exactly the cells first find out which is bacterial part of the proteins or DNA or whatever they will find, and what is our self materials inside. You know, if you look at the proteins, the structures of proteins are very similar, that bacteria use, that we use, very similar in many aspects. So it's kind of difficult to find out. So the first thing, there are some cells out there present in our tissue all the time. They're circulating the bloodstream. They find out if there is any infection. Now for example, say there is a damage in our skin, through that some bacteria take entry into the tissue associated with the skin. So there's a entry of the bacteria out there. So once those, so let me draw the idea. This is the, let's say, the vessel, blood vessel, and this is the associated tissue where we have the damage. There is the damage in the blood vessel or somewhat like that, or now that damage is clot, like that damage is filled with the clot, but still bacteria take entry, and let's say these are the bacteria. Now as bacteria take entry in the tissue, and we know all the time our body is prepared to fight against infections, that is known as the innate immunity. There are cells that are circulating the tissue and bloodstream to fight against those infection or infective agents. So the example of one such thing is macrophage. Sorry, let me change the color here. One example is the macrophage. Let's say this is the macrophage, which is a phagocytic cell who stays there in the nearby adjacent tissue. There is also other cells called dendritic cells, like that. So these dendritic cells, macrophages, they are present in the adjacent tissue where the bacteria take entry. At that condition, this macrophage will engulf this bacteria inside via the process known as phagocytosis. Okay, so once they engulf this bacteria by phagocytosis, they will start releasing certain chemical factors in this nearby tissue, and that chemical factor that is released in this nearby tissue includes, they are known as cytokines. They are known as cytokines. So as they release these cytokines to the nearby tissue, those chemical factors further signal other cells to come and join, lymphocyte cells to come and join, all those white blood cells to come and join in this area, because you know white blood cells are circulating through the blood vessel. So if this cytokine released and go to the blood vessel, then they can bring some white blood cells to the area and start the action in more amplified fashion. Now my question is, how do these cells recognize these pathogen factors? Now for recognizing pathogen factors, there are specific unique sequences that these cells are looking for, which is unique to bacteria or which is unique to a virus, but it should not present in our own body cells. For example, you know, there are different body parts of bacteria. Now, if they engulf the whole bacteria, that is another thing, because you know, whole bacteria, it is also circulated, covered with so many lipopolysaccharide things, if it's a gram-negative bacteria. If even it's a gram-positive bacteria, there could be proteoglycan layers, specific proteins in the surface of bacteria that can be a signal for the macrophage or dendritic cells that this is a foreign particle, and they will engulf that. But let's say there's a fragments of bacteria also that is present in this area, fragments that are present. Now that fragment could be a body part of bacteria. Okay. Now the body part could be, let me tell you, the body part could be flagella, or the protein fragments that make flagella, which is flagellin. It could be lipopolysaccharide, which is a prime endotoxin part of the gram negative bacteria. Okay. And also there are different proteoglycans or glycoproteins found on surface. For example, diacyl lipopolysaccharide, a lipopeptide, you can say, not polysaccharide, it's a lipopeptide, triacyl lipopeptides. These are some things which are very unique to only bacteria, that are not present in our eukaryotic cells, usually not found. So whenever they find these things, flagella fragments, LPS, that are involved with this nearer region of the tissue, find yes, there is something wrong, this thing should not be present in the tissue, so definitely something invading us. So let's find that out, and let's bring some more people into this place and start to find and search. That is the idea here. So immune system always work, like it's not going to take it the activity on its own hand. Even macrophage is a scary cell. It can damage so many other cells, like so many bacteria, and they can engulf it, damage them, break them down, but still they will call upon another much specific line of defense known as acquired or adaptive immunity. So they need adaptive immunity to boost this whole scenario, and for that they need to secrete some chemical factors known as cytokines, and first of all they need to figure out which cells we are talking about, or which identifying materials like proteins or DNA fragments we are talking about. Now one such identifying things, now those all things which are very specific to bacteria and viruses only, or pathogens only, known as pathogen associated molecular patterns, PAM, pathogen associated molecular patterns. Okay. So once this pattern means, there could be some sort of LPS, flagella, or as I told you, like fragments of their like diacyl glycoprotein or lipoprotein or triacyl lipoprotein. These are the things that they are searching for there. These are the PAM. Now this PAM could be recognized by several different receptors that are found in our immune system cells. For example, the cells like macrophage, cells like dendritic cells, they can find these PAMs with the help of some receptors. And there are three types of receptors that I want to talk about here. One is the toll-like receptor, or TLR. Toll-like receptor. Toll-like receptor, or TLR, is one of those kind of receptor that can detect the presence of all those materials or bacteria specific materials in the cell, in the tissue. Now as you see, there are different varieties of TLRs that are found, and each of those TLR and set of TLR molecules, they figure out a specific type, they identify a specific type of bacterial component. Okay. Another type of receptors that we are talking about, three different types. One, we talk about TLR as receptor, second is a nod, and third one is RIG1 or RIG-I, must say. So these are the three types of receptors that we want to talk about, which are involved in the detection and identification of pathogen. Now TLR, a toll-like receptor we talked about, We'll talk about that in much more details, because there are different types of TLR for their function. NOD is nuclear oligomerization domain family of proteins. Now in this case, they are nuclear oligomerization domain proteins, and these proteins or NOD proteins, they actually find out the proteoglycans that are delivered or that are present in the surface of a bacteria. For example, muramyl dipeptide. This is one of the example of a proteoglycan that is found in pathogens, in the surface of the pathogen. Okay, so these NOD family proteins find out those surface peptidoglycans or proteoglycans that are present. And the third kind is this RIG. Now RIG type of proteins, they detect the double stranded RNA that is present. Okay, because double stranded RNA is not a common feature of eukaryotic cell. Even if it's produced, it's degraded. But normally, if virus infects our body, they can carry double stranded RNA as the genetic material. So if we find double stranded RNA circulating in the tissue, that means a virus is definitely infecting us. So we can find that out. So these are the three different type of receptors that is there. Now most of these toll-like receptors, they are membrane associated receptors. That means they are embedded in the membrane. Okay, so they always attached to the membrane, and they are leucine rich proteins. Okay, a specific receptor structure with more leucine amino acids in the protein, and they are always associated with the cell membrane. So either these toll-like receptors, if I draw the picture here, either these toll-like receptors can be found on the surface of the cell. Let's say this one. Either they will be found exactly the surface of the cell membrane, or sometimes what happens, even inside the cell there are vesicles formed, where inside the vesicle there will be the fragments of the bacteria and the pathogen. They can also be present inside that vesicle. Okay, but ultimately they will be embedded with the membrane. That is the idea. Now you see here some of this TLR that we listed, TLR 1, 2, 4, 3, 5, 6, 7, the different types. Now TLR 1, 2, this is acting as a dimer, and they actually help in the determination of or on the identification of this triacyl lipoprotein or lipopeptide. This is the idea of TALP for the TLR 1 and 2. For TLR and 3, they go for diacyl. Okay, TLR 4 recognizes LPS or lipopolysaccharide layer, which is a prime identifying thing for gram-negative bacteria, because all of the gram-negative bacteria carries that. TLR 5 recognizes flagellin. Flagellin is the unit protein that is combined together to make flagella for bacterial flagella, remember. Because in eukaryotic flagella it is made with tubulin protein, but bacterial flagella is produced with flagellin protein. And then TLR 3 and TLR 5, both of them, they recognizes RNA, and these are some intravesicular TLR. They are present inside, like embedded inside this membrane a little bit, inside the vesicle, and they find out either single-stranded RNA or double-stranded RNA. So once they find the single-stranded or double-stranded RNA, that can give us some proof of what kind of a virus and bacteria that are infecting us. So that is the job of TLR, and TLR is one of the most important of the receptors among these three, probably the most important one. Second, I told you about the and the function of TLR is that TLR not only recognizes the type of molecular pattern. Once it recognizes the pattern, they signal through the downstream signaling process is done, and then finally activates the transcription factors. Once the transcription factors are activated inside the nucleus, then these transcription factors help transcribing so many interleukins. Interleukins are cytokine molecules. So they will start producing more and more cytokine. And remember, what is the importance of cytokines again? It's just like the phone, molecular phone. So as they produce cytokines, it can call upon many more more cells of our immune system, and the cytokines actually help leukocytes or white blood cells to roll along this vessel and come out of this vessel to the infected tissue. So cytokine generation is very important for keeping this reaction to the next level and forward to the more bigger level or bigger scale. That is the job of TLR. NOD type of proteins, they will also do the same type of thing. They also signal, and ultimately the signal is ultimately provided to the nucleus inside the cell, and the nucleus, there are some transcription factors activated. They will again type cytokines. Cytokines example, interleukin 1, interferon. Okay, these are the cytokines that we are looking for at the very beginning. So interleukin 1, interferon, IFN, type 1, and so many different types of interleukins are generated. Interleukin 8, which will be required for the process of leukocyte rolling and adhesion. All these interleukins start to be released. And again, last one, this RIG, it functions a little bit different way. While TLR and NOD directly provides the signaling and it activates the transcription factors, but RIG ultimately provides some. So if they find out that double-stranded RNA is present and if the cell is damaged properly, there is no way for the cell to live and do the rest of the job. In that case, they will also recruit other apoptotic factors to kill the cell. Or this RIG will ultimately help in the activation of or overexpression of interferon type 1. So once it starts producing interferon type 1 as a cytokine, that interferon type 1 cytokine helps to bring more and more of this macrophage, more and more dendritic cells to the place, and also it will also help the process of bringing the leukocytes from the vessel to the infected area of the tissue. So that in a sense is how pathogen is recognized, and it's not done because once you recognize the pathogen, you need to do the signaling. You need to bring some more cells so that some more pathogens could be detected, and also you need to bring lymphocytes. You need to bring all those cells that will be required for developing the acquired immunity or adaptive immunity, because that is how the immune system works. But remember, once they recognize all these things, this macrophage, dendritic cells, the number of macrophages will be higher because more and more macrophage will come there, more and more dendritic cell will come in the infected place. And also they will signal these interleukins which will help white blood cells to come and squeeze through this vessel wall to the infected area of the tissue. Once they are squeezing through that place, so you see a lot of things will be going on in the infected tissue. More cells are rushing in. Okay, so as the cells are rushing in, that part of the tissue start to swell up, right? They start to release all those interleukins, they start to release some histamines, bradykinins, and all those different chemical factors that is doing the job of vasodilatation. Vasodilatation means the dilation of the blood vessel. As well as all this process start to take place, heat start to radiate and generate. And as all these things going on in the tissue, nerves kind of getting damaged because of these infections and all the process going on, so you will feel pain. So what things you are noticing now? You feel pain in your wound, you feel swelling, you feel redness because obviously all those red blood cells are gone and clotting start to form. Redness, pain, swelling, and radiation of heat. All these things that we are looking are the prime sign of a process known as inflammation. And inflammation is a response of your body which are telling you that we are fighting against the infection. So whenever you see there is an wound and you find something like pain and swelling and redness, all stuff, that means it's good, it's for your good. Your body is preparing itself to fight against the infection. That's why the place becomes swelled and all the stuff is going on, because your body have an active immune system. Okay, so that is the science of inflammation, and inflammation is the integral part of innate immunity. While not only the cell but also some other factors will come so many times later, like the humoral part of the factors will also come. For example, the complement system, which we are not talking right now, we'll be talking about that one video later. You can see that complement system will take their activities as well, but before that, this is the prime and first level of defense that is going on in this site of infection. Okay, so that in a sense is a process of pathogen recognition and how inflammation is related to that. I hope this video helps you out. If you like this video, please hit the like button, share this video with your friends, and subscribe to my channel to get more videos like that. Thank you.